Okay, good morning. We have a pretty fair number of people here today, so we're going to go ahead and get started. I hope to finish chapter 2 finally today on chemistry, which means that we can open up a test on next week, a lecture test. I want to wait till the lab test is done before I open up the lecture test. So it'll probably open up next Wednesday, maybe 22nd. I have all these lab exams set to end on the 21st, and we can open up the lab exam, lecture exam on the 22nd, let it run for 10 days. And I think we're gradually getting all of our lockdown browser issues fixed. If you have one, let me know. I've got our computer specialist at Severe looking into some of the problems that we can't figure out. So we'll just go from there. Anyway, anybody have any questions on anything? Okay. Then I'm going to go ahead and open up the screen and try to get into chapter 2, and we'll go from there. There we go. I think we had stopped somewhere around protein structures, and we had talked about the shapes. This is a good place. We'll start out here with fibrous and globular proteins. Now, proteins, this is the third of the 4 organic molecules. Proteins come in 1 of 2 categories. They're either going to be a fibrous or a globular protein. I like the name globular. It sounds messy. So, fibrous proteins are the structure proteins. The things like muscle cells are filled with fibrous protein. The protein in our tendons and our ligaments, all that collagen fibers are structural proteins. They are all insoluble in water. They all are arrayed in strands side by side. Remember the imagery from the lab of the collagen fibers? You know, the woven strands side by side by side by side. That's what fibrous proteins are like. Muscle cells are like that. Most of them are either going to be tertiary or quaternary. They have great strength. Individual strands of collagen fibers are stronger than individual strands of the same size of steel. And you know, a protein like a tendon made of collagen fibers will snap the bone when it's stretched faster than the collagen fiber will break. In other words, if we're stretching a tendon, it's more likely to break the end of the bone off that it's attached to, then the tendon will snap. And we'll talk in AP2. We'll talk why that doesn't always work for the Achilles tendon. Anyway, keratin, waterproofing compound in our bodies. Elastin, elastic fibers in our skin and our cartilage lets us, and our arterial walls that lets us to rebound and expand and contract and resume our shape. And of course, collagen, as it says, the single most abundant protein in the body. It is all very, very strong. It is what we find wherever we want to have strength, where we hold joints together, where we attach muscles to bone, where we contract our muscle cells. The contractile fibers, the fibers within our muscle cells, are all fibrous protein. The globular proteins are different. These are the enzymes. These are the hormones. These are the antibodies. They are very sensitive to temperature. They're very sensitive to changes in pH. They are soluble in water. They have a problem trying to get into cells because they are soluble in water. Only the the only let me rephrase that. These protein-based hormones are water soluble. They have a hard time entering cells. The fat soluble or the fat-based hormones based off of cholesterol, like testosterone and estrogen, slide right through the membrane. But water-soluble proteins have a real problem. So many of these are going to be functioning as antibodies. Antibodies glom onto foreign structures and are targeted for destruction or removal. Many of these function as hormones, which are chemical messengers sent out by the pituitary gland, telling a cell to do something, like tell the thyroid gland to make more thyroid hormone, or tell the kidneys to make more erythropoietin to stimulate red cell production. These are messengers sent by these are chemical messengers sent to the cells to tell them to do something. Tell the kidney to make more erythropoietin so the bone marrow will get instructions to make more blood. Okay, that's a role of a chemical messenger. Molecular chaperones, not what it sounds like. Molecular chaperones are there to actually twist proteins into their specific shapes. And of course, enzymes. Enzymes go back to the whole idea of, the question, thank you, Laura. Lecture test due on the 24th, that's wrong. Everything is sliding back a little bit. We've got plenty of time. I'm going to slide it back, the due date, I'll slide it back to at least to the 30th of September. So I'll have to look and see how far back I want to send it. But I want to make sure I want to make everybody done with the lab test before they start on the lecture test. So yeah, I'll change that date in e-learn. So anyway, thank you for bringing that up. I appreciate that. Okay. Enzymes, one of the most common form of globular proteins, function as a biological catalyst in our bodies. An enzyme is a molecule that simply functions to bring other molecules together to cause a reaction to occur. Now, we know that in chemical reactions, we can increase the rate of a reaction by increasing the temperature, by increasing the concentration of the reactants, and shrinking the particle size down to very, very tiny particles so they react faster. We can't do any of that in our bodies. We are stuck pretty much at 37 degrees C, 98.6. The only time we raise our temperature is when we have a low-grade fever, and that's just a very few degrees, and that does work to enhance the breakdown of bacteria, but we don't do it very often. So we can't raise our temperature. The concentration that we're dealing with in our bodies for reactions is very, very small to begin with. The particle sizes are very, very small. So we can't do anything about that. So the things that we would do in a lab, add some more sodium bicarbonate, add some more vinegar, heat it up. None of that stuff works. None of that stuff is going to work because we can't do it in our bodies. So we use enzymes. Enzymes are a catalyst. They make reactions go faster without any damage to the enzyme. They can be reused over and over and over again. What they do to make a reaction work faster is simply bring the reactants close together. See, a normal chemical reaction depends on two reactants bouncing into each other and then a reaction occurring. See, every particle is going to be in motion. Particles in solution are in motion. Particles in the air are in motion. Even particles in a solid or a gel are in motion. A little slower, but they're still in motion. Reactions occur when these particles randomly run into each other. That takes a lot of energy to finally find if you're particle B and you're looking for particle A, it takes a lot of random energy to bouncing around until you finally hit particle A and you can react. And then you get a product. Well, what an enzyme does is it floods the area where a reaction is taking place with lots and lots of copies of itself. The enzyme is designed to hold on to particle A and particle B and bring them close together. That's all it does, it brings them close together so a reaction can occur. So we flood an area with enzymes, a specific enzyme, then particle A and particle B will be brought together sooner with less energy expended. And so we can't do anything about temperature or particle size or concentration. We can produce lots of enzymes. And they make reactions work by simply bringing the particles together. That's all they have to do. And this is an example here. We're looking at a molecule of starch. And if we bring in an enzyme called amylase, salivary amylase is found in our mouth. It's an enzyme that's in solution in our saliva. And what it does is when we eat a carbohydrate full of starch, the amylase will glom onto the starch and break apart two molecules of glucose linked together as a disaccharide. In this case, it's forming maltose, malt sugar or maltose. So we take the large carbohydrate and we break it into smaller pieces. This is the first step in digestion. Digestion starts in our mouth, and we use amylase. It's the only place amylase works is in our mouth. And it will start chopping up starch into much smaller molecules of maltose, malt sugar, if you will. It's often said that you can take a cracker and place it in your mouth and let it dissolve. You would eventually taste the sugary sweetness of the maltose. Usually what happens is by the time the cracker is so dissolved like that, you end up swallowing it. So I've never been able to get it to work, but theoretically it would make sense that as the starch in the cracker is being broken down, you would eventually start tasting the disaccharide sugar. So, but the amylase is an enzyme. It has an ASE ending, and it will cause a reaction to occur. It will cause this reaction to occur. It will bring the particles together to cause a reaction to occur. Now, the problem that we run into in our bodies is that these globular proteins are very sensitive to temperature and to pH. When there's a process here called denaturation. They become denatured. If you are a globular protein, when you are exposed to changes in temperature or pH, you unfold, your shape changes. The three-dimensional shape of these globular proteins is very, very specific. The shape of the active site where the reactants land. The reactants are called the substrates. The shape of the active site is very specific. It's like the lock, looking for a unique key. The substrates, the reactants, have a very specialized shape that can land on the active site, on the enzyme. The active site doesn't have that shape anymore. It's not going to recognize these molecules. They're not going to be able to land there, and we aren't going to be able to cause a reaction to occur. Either building something up or tearing it down. Fibrous proteins don't have this problem when they're exposed to higher temperatures or lower temperatures or changes in pH. They become more stable, but globular proteins become deactivated. A rise in temperature, a drop in temperature, a rise in pH, a drop in pH. Now, if it's a minor change, we can reverse it. But if it's a major change, we can't. Look at the egg. If you cook an egg, if you fry an egg, the white of the egg, when you crack that shell open, that white of the egg is transparent. It's sort of pale yellow. It is made up of albumin. It's albumin. Albumin will turn white and solidify. It was a gel, sort of like a sort of semi-gelatinous, goopy mixture, semi-gelatinous solution with the yolk sitting in the middle of it. But if you apply heat to it, it solidifies and turns white. You can't go back. You can't take the egg here and undo the denatured change that took place. The proteins have changed. They've altered their shape. They have become twisted and bent. And now you have this solid that not only is it solid, but it's also white. It's no longer the transparent gel that it was. You can't reverse that. Active sites on enzymes become deactivated. Proteins can't, proteins change their shape and no longer fit where they're supposed to go when they become deactivated like this or denatured like this. And in our bodies, we are very concerned when, when an individual experiences a cardiac arrest, that we can't, the cells, the enzymes in the cells that are maintaining homeostasis cease functioning when the heart stops. So cells don't die immediately, but cells start dying in 5 minutes. Brain cells die in 5 minutes. Muscle cells die in about 10 to 12 hours. Heart muscle takes about an hour to die. But as those cells are running out of oxygen, their enzymes are breaking down. So they can't do the reactions they're supposed to do. An enzyme, we say it's a biological catalyst. Here's a couple of points about the enzyme. It causes a chemical reaction to speed up without any change to itself, simply by flooding an area. If you have reactant A and reactant B, and you want to bring them together, and you can't wait for them just to randomly bounce into each other, what will happen is if you flood the area with an enzyme designed just for reactant A and reactant B, it will encounter them. It will encounter the reactants drifting around in solution or in the air or wherever, and hit into them and bring them together. Physically bring them together. That's all they do, is they physically bring the reactants together. The reactants are called the substrates, and the substrates land on what are called the active sites on the enzyme. There's substrate 1 and substrate 2 coming together. They form the enzyme substrate complex. The reaction then occurs, and we have a product that we let go, and the enzyme goes about its business and goes out and gets 2 more reactants of the same type. And so instead of random reactions taking place, we have millions of reactions taking place. The enzyme isn't effective. The enzyme is used over and over and over again doing its job. So since we can't affect size, temperature, or concentration, we can use enzymes to make reactions work. But if our pH changes, we can't do this. If our pH changes, things like this can happen. The enzyme can become denatured. See, this is what happens with a regular enzyme. The reactants land on the active site and they react. They give you the product. But if you alter the shape of the enzyme with temperature, it's denatured. The active sites are damaged or destroyed. And so the reactants can't fit there anymore. You won't get the reaction. In our bodies, if we raise our temperature, if we raise or lower our pH, the same thing happens. and the cells stop functioning. A person who experiences cardiac arrest is no longer pumping blood around to the lungs. And why are the lungs important? Well, so we have to breathe. However, the other thing we do with the lungs is we send all of our waste carbon dioxide to our lungs. Every pass through our bodies only use 20% of the available oxygen that's in blood. There's about 80% reserve left over. So we've got, for about at least 5 minutes, we've got plenty of oxygen, at least for as far as the brain's concerned. Our cells are drawing on that oxygen to stay alive. They're busy generating carbon dioxide. That carbon dioxide is being released into our blood, which is not moving. That blood would normally be going to the lungs to be reoxygenated and also to get rid of the carbon dioxide. See, and that carbon dioxide that goes into our blood is converted to carbonic acid. That's how the easiest way to transport it. We convert it to carbonic acid, then when we get to the lungs, we convert it back to CO2 and we breathe it out and get rid of it. You've heard me say over and over and over again that carbon dioxide drives our breathing rate. Well, it's because of the change in pH. As carbon dioxide levels go up, we become more and more acidic. So we breathe out. We breathe faster to get rid of the CO2. I've said that a million times to you. What happens in a case where a person's heart has stopped, they're not pumping any blood to the lungs. The carbon dioxide is not getting removed. More and more carbonic acid is forming. It's getting more and more acidic inside the plasma, and it's affecting the cells. The cells are becoming more acidic. The enzymes inside the cells are becoming denatured. Their shapes are changing, and the globular proteins stop functioning. And so all of these occurrences are taking place, and the patient's dying. They're not dead when the heart stops, but they're going to be brain dead in about 5 minutes. And then other cells are gradually going to be dying off too. So this is one of the reasons why during a cardiac arrest, usually a patient is administered sodium bicarbonate solution, good old baking soda, administered into the plasma to start neutralizing the production of carbonic acid as you attempt to resuscitate the patient. Once these enzymes are permanently denatured, they're not going back. And so we see significant damage occurring to our cells if we can't stop that problem. Let me go back to it. So what about enzymes? Well, they usually have two parts to it. They have a protein part called the Apoenzyme, and they have a second part that's either a cofactor or a coenzyme. If there's metal involved, it's a cofactor. If there is a vitamin involved, it's a coenzyme. A coenzyme is not a wannabe enzyme. It's not an assistant enzyme. It's not a type of enzyme. A cofactor simply allows the enzyme to do its job, do it better. But it is not, the vitamin is never going to become an enzyme. It just helps the enzyme work better. Enzymes are very specific. They act on very unique substrates. They will only work on, starch or on, carbonic gases, carbonic acid. They'll only work on that. They'll only work on different types of substrates. They are substrate specific. Amylase, the enzyme amylase, will not work on carbonic acid issues. Amylase only works on starch and nothing else. Other enzymes, whether they are a catalase working on hydrogen peroxide or a hydrolase working on something with hydrogen attached to it, those are specific. And enzymes, whenever you see the ASE ending, on the name of the protein. It's an enzyme. ASE signifies it's an enzyme. I don't know why, that's just the nomenclature. So oxidase, hydrolase, catalase, catabolase, any of these are all types of enzymes. Dehydrogenases, something that strips off hydrogens, whatever it does. The ending, the ASE indicates it is a enzyme. And I showed you, again, I showed you breaks. Let's go forward. Okay. Enzymes lower the activation energy of a reaction. They do this because a reaction can either go, an unassisted reaction depends on the reactants slamming into each other as they randomly move. And that takes time. If you have small concentrations and small particles and lower temperatures, it's going to take time for these reactants to slam together and cause a reaction. That's what they do. They physically come together and a reaction occurs. But by flooding an area with millions of enzymes, these reactants don't have to waste all their energy bouncing around, hoping to run into each other. If a reactant A and reactant B are randomly bouncing around, trying to encounter each other so they can produce a product, it may take a long time. It may take a lot of energy until they actually slam together. But the enzyme will grab the A's, will flood the area with millions of enzymes and grab all the particle A's and all the particle B's and bring them together, and they'll react, and then they'll kick that product out, and they'll go do it again and again and again. So they lower the energy required to have a reaction take place. And they get to do it over and over and again. So they lower, this is an important point here, they lower the activation energy of a chemical reaction. I want to circle that on your handout, on your printout, on a PDF. Enzymes are biological catalysts. They lower the activation energy. If this works, you can see
and enzymes.
If you couldn't hear it, then you can at least bring it up on your own PowerPoint and watch it. So if you did hear it, every cell has enzymes. Every cell depends on enzymes for chemical reactions. It can bring reactants together and produce a product. It can take a product like starch and break it apart into smaller components. It can work either direction. We depend on enzymes constantly. Since, again, we are stuck, we are limited to the small concentrations, the set body temperature and the small particles. We can't do anything about any of that. We can produce lots of enzymes. Now, this is a good little example here. Here we're linking amino acids together. Amino acids are the components that make up proteins. And so an enzyme can take two random amino acids that fit in its active site. They're not just so random there, they have to fit there. And so the enzyme will take the amino acids that fit their active site, bring them together, allow a bond to form, a peptide bond, and then release the product, and then go do it again and again and again. And this is how we link. This is the basis of how we link amino acids together to make proteins. I showed you that already. Okay, so talking about amino acids and proteins, let's talk about the instructions that we follow to link amino acids together. The nucleic acids are the fourth category of biological molecules. They are also made up of carbon, hydrogen, oxygen, and nitrogen, and phosphorus, just like the proteins are. The polymers of the nucleic acid polymers are made up of monomers called nucleotides. And these molecules, these polymers of nucleotides, are the largest, longest, I tripped over that word last time. They're the largest molecules in our bodies. A DNA molecule, one of the nucleic acids, a DNA molecule can be over 6 feet in length. They're all made of these nucleotides. A nucleotide has a base with nitrogen in it. It has a 5-carbon pentose sugar, and it has a phosphate group attached. So a nitrogen base, a pentose sugar, and a phosphate group. This is what a nucleotide is made of. The two classes of nucleic acids are the deoxyribonucleic acid, the DNA, and the ribonucleic acid, the RNA. The difference is that the DNA has one less oxygen than the RNA. That's why they call it deoxyribose nucleic acid. You kicked out an oxygen somewhere. So these are what the nucleotides look like. There is the phosphate group right here. There's the 5-carbon sugar, the pentose sugar. Pento means 5. And there is the nitrogenous base. So you have the sugar, you have the phosphate, the sugar, and the base. The phosphate, the sugar, and the base. And there are 5 different bases that we encounter. So this is what the nucleotide, how it appears. The phosphate, the sugar, and the base. These, the nucleotides all have the same basic structure. Now the DNA, we've all heard about DNA. DNA is our genetic code or genetic blueprint. It has all of our genetic information available to us. It is the master cookbook to make all of the proteins in our bodies. It is essentially essentially has all the instructions we need to make everything about us. It's all contained in the DNA. It's what we call a double helix. It's like a double spiral staircase wrapped around itself. Or if you imagine, take a long ladder and twist it, and if the ladder was flexible, if you could start twisting the ladder into a spiral shape, and you would have 2 rings all the way around in this double helix. The nucleotides in DNA have a deoxyribose sugar, a phosphate, so meaning it's missing an oxygen. It has one less oxygen than the ribose sugar. It has a phosphate group, and it has one of the following bases. It's either got a purine, such as adenine or guanine, or it has a pyrimidine, and it has a pyrimidine, such as cytosine or thiamine. Okay, now that I've said purine and pyrimidine, you can probably forget about hearing those again. Anyway, the 4 bases are adenine, guanine, cytosine, and thiamine, and they bind to each other through base pairing. They have complementary bonding, meaning adenine will always bind to thiamine, and guanine will always bind to cytosine. Hydrogen bonds form across the DNA strand between the adenines and the thiamines and the guanines and the cytosines. So it's what we call a complementary base pairing. Adenine always pairs with thiamine. Guanine always pairs with cytosine. Now, and I'll show you at the end of chapter 3 that sometimes they don't. Sometimes mistakes happen. The error rate in our DNA is about 1 in a billion. Sometimes cytosine does pair with thiamine, and adenine pairs with guanine. Mistakes happen, but not very often. Again, with an error rate of at least one in a billion, that's not bad. So, but normally we have complementary base pairing. We can pretty much say adenine always pairs with thymine, guanine always pairs with cytosine. This is the double helix strand. It's like a spiral staircase, if you will. The strands are held together, or they appear to be held together by the rungs. The rungs are the base pairings, the complementary base pairings, adenine pairing with thymine, guanine pairing with cytosine. For example, there's adenine in red. It really isn't red, but there's adenine pairing to thymine. There is guanine pairing to cytosine. And again, there's a guanine, there's a cytosine pairing to guanine, there's a guanine pairing to cytosine. So there's a thymine pairing to adenine. Adenine and thymine always pair together. Guanine and cytosine always pair together. They are held together by hydrogen bonds that form between the bases. So if we go back here and look at the nucleotide, there's the base right up here. There's the phosphate, there's the sugar, there's the base. And as these bases line up across from each other on the double strand, hydrogen bonds form from the adenine to the thymine, from the thymine to the adenine, from the cytosine to the guanine, and the guanine to the cytosine. And those hydrogen bonds that form across the bases are the cause of why the molecule forms its spiral structure. The twisting caused by the hydrogen bonds forming caused the molecule to change its shape from a straight line structure into this spiral, this double helix, simply by the pull of the hydrogen bonds. And that gives us this double, what we call the double helix. Now, RNA is a little different. RNA is a single strand molecule. It isn't double stranded. It still uses nucleotides, but it uses the ribose sugar instead of the deoxyribose. So it doesn't have one oxygen less like the DNA does. It has so it has ribose. Ribose is a 5-carbon sugar. Deoxyribose is a 5-carbon sugar. This means that it's missing an oxygen. There are 3 types of RNA, and RNA does all of its work outside the nucleus. Now, the DNA never leaves the nucleus. The DNA always stays in the nucleus and lets the RNA do its work. So the DNA is the control center and sends its instructions out using RNA. And there are 3 types of RNA. There's what's known as messenger RNA. There is transfer RNA. And there's ribosomal RNA. Messenger RNA takes the instructions out of the nucleus. Transfer RNA starts to assemble proteins by bringing amino acids together. And ribosomal RNA, it helps to make the site of protein assembly form. So, and here's the difference. If you can't hear this, I highly recommend you watch this on your own PowerPoint. DNA and RNA Now, a gene is nothing more than the physical location of the instructions on a DNA molecule. That's all a gene is. Now, when we say here that the sequence of the bases is significant, the order of the bases specified in the DNA determines what amino acids are going to be used to make proteins. So these instructions in DNA, our DNA code is a list of instructions on the order of the amino acids we bring together to make a protein and what amino acids we bring together. So in a lot of ways, it's like a cookbook. It's like if you're gonna, the instructions on how to make a cake. The instructions are going to tell you to mix eggs, water, oil, and flour together, and then place it in the oven at 350 and bake it for however long. But if we didn't have those, and those instructions are pretty specific, you don't stick the cake mix into the oven first and then mix it together. The instructions specified in the DNA at the gene tell us how to make the protein. Whether it's a protein for hair color, eye color, skin color, dangling earlobes, believe it or not, earlobes, whether they're attached or they dangle, is controlled by our DNA, along with all the somewhat more important traits, like blood typing, for example, or physical appearance or body stature. All those are controlled by our DNA. And the DNA, how it controls that is by the order of these nucleotide bases. These bases are grouped together in clusters of 3. Each cluster of 3 is called a codon, and each codon is unique for an amino acid. So since proteins are made of amino acids in a specific order, the DNA gives us that order for each protein. Sometimes mistakes happen. Sometimes we make hemoglobin, and I'll talk more about this later on, but again, with sickle cell disease is a genetic disorder caused by the substitution of the wrong amino acid into the number 6 spot on a molecule of hemoglobin. Hemoglobin at the number 6 spot should have glutamic acid. It's like 360 amino acids long. Number 6 spot has glutamic acid has supposed to have glutamic acid, but in sickle cell hemoglobin, it has the amino acid valine. Valine's a perfectly fine amino acid. It just doesn't belong in the number 6 spot in hemoglobin. So a mistake is made with the hemoglobin by putting in the wrong amino acid. And it's because the instructions are wrong. A person who has sickle cell disease has received a gene for hemoglobin that is defective because it produces the wrong type of hemoglobin. No fault of their own. There's nothing they can do about it. There is therapy for it, so it's not going to correct itself, and it's through no fault of their own. They are now either a carrier or they express the disease simply because of one little mistake, one little amino acid that is slightly different from glutamic acid inserted in the wrong place, and it affects the whole makeup of their blood. So these are what genes are. Genes are nothing more than the physical location of this information. So the gene for hair color, the gene for eye color, physical location on the DNA. Now, the good news is, for things like that, is that, well, the bad news is the mistake for the incorrect DNA for hemoglobin, for sickle cell hemoglobin, is going to show up throughout the DNA strand. Because we have multiple locations of the same genes. We have redundancy. We don't just have one spot and one spot only to make hemoglobin. Unfortunately, with sickle cell disease, they're all defective, or most of them are anyway. However, hair color, eye color, skin color, blood typing, those genes occur in multiple locations in our DNA strand. So if there is a mistake that occurs spontaneously, we can ignore it and keep on going. Because mistakes happen all the time. But we have so many genes that are identical. So if we have a mistake on blood type on gene number 375, well, we've got another 5,000 genes that are fixing that. And so the defective gene never gets a chance to make a bad protein. Unfortunately, with sickle cell disease, that's not the case. So we don't put all of our genes in one location only. We have many locations throughout our DNA. And each strand is 6 feet long. So, okay. More on that when we get into chapter 3. Now, the third type of nucleic acid here is based on something called ATP. And we've talked about DNA and we talked about RNA. They're both made of nucleotides. DNA is double-stranded. RNA is single-stranded. Now we're talking about a compound that is just the nucleotide. It is similar to the nucleotides in RNA. It has a base, it has a sugar, and it has a phosphate group. But here, ATP has 3 phosphates. We call it adenosine triphosphate. This is what we use to transfer energy around our bodies. The energy that we get comes from glucose. The energy that we get in our bodies comes from glucose. We oxidize glucose in our cells, and when you oxidize something, you release energy, you release electrons. Okay, so we're releasing energy from glucose. We are oxidizing glucose. That energy and electrons are captured by adenosine triphosphate. And then we use that ATP to take that energy to a specific location in the body and use it there to do something. ATP provides the power in our cells for things to happen. Muscle contractions, nerve impulses, pumping substances across a membrane. Anything that requires energy, we get our energy from ATP. So we're taking the energy stored in glucose, transferring it to ATP, and using that to do things. Now, that's the good news. The bad news is that we can't store ATP. In fact, ATP only lasts for about 7 seconds. That's all. But we never run out because we're always making more ATP. I shouldn't say we never run out. At some point when we're dead, we run out. But we essentially don't ever run out of ATP. We are busy generating new ATP all the time. We're taking the energy from glucose and putting it into the adenosine triphosphate molecule. And that powers our activities. It only lasts, we only have it available for 7 seconds, but we're constantly making it new. So we're constantly refreshing it. We can't store that energy as ATP. We can only store energy as a fat or as a carbohydrate. So this is what the ATP molecule looks like. It has the adenosine base. It has the ribose sugar, the 5-carbon sugar. It has 3 phosphates. ATP has 3 phosphates. The 3rd phosphate is held onto the nucleotide by energy. Energy from a chemical reaction, usually the breakdown of the oxidation of glucose. We have what's known as a high energy bond that occurs between phosphate number 2 and phosphate number 3, because there's 3 phosphates there. So between phosphate number 2 and phosphate number 3, when we apply energy there, we can force the 3rd phosphate on. We go from ADP, adenosine diphosphate, the low energy version, to adenosine triphosphate, the high energy version. Granted, it only lasts for 7 seconds. That's all we need to transfer energy to, a nerve impulse or a muscle contraction or something like that. So it is the conversion of ADP to ATP that allows us to store energy in that high energy bond for 7 seconds. That's what we do. When we break the bond, when we break that bond, we release that energy to do work. And then we end up with what we call an inorganic phosphate and adenosine diphosphate. See, there's the bond. We break the bond. We have the inorganic phosphate here and the adenosine diphosphate here. And we can reload that bond by applying energy to it from the oxidation of glucose. We park it in there, we transport it somewhere in our body, and we use it to do work. So while we get all of our energy from glucose, the transport medium, the currency, if you will, the money that we pay as a cell goes, is the ATP. We use the ATP to transfer this energy around. And so we're always making ATP. We can never store it. It will only last about 7 seconds. Okay. And that is the end of chapter 2. So we are done with chapter 2. And as I said, I'll probably open up the test next, whatever day, the 22nd. I guess that's Wednesday. So I'll let it run. I know the e-learn said the test was going to be the 24th, but I will tell you, having taught A&P for 18 years, you never know when you're going to get to when you have to a test date. You just don't know, because you don't know how the lectures are going to go. So anyway, test will be we'll open up on the 22nd. It will cover chapters 1 and 2. And there are study guides available, but I'm not going to open it up until the lab test ends. So the lab test will end on the 21st. And I have 10 after 12. I probably can't get anything started. So I'm going to just call it, I'm going to let you out early and call it a day here. We've covered a lot today. And so we'll pick up on Tuesday of next week and go right into chapter 3. Question over here. Let's see. Practice test and actual test, are they similar? Yeah, they're similar. They're not the same questions, but they're the same types of questions. I mean, I'll tell you that there are only so many ways you can ask the same question. So, and the practice tests were real exams from about 8 years ago, so they're different. I don't think you're going to see any of the same questions on there, but you're going to see same types of questions. I highly recommend taking the practice test, at least just to get a flavor of what my exams are like. I try very hard not to have any. You'll never have a trick question. I try very hard not to have an ambiguous question that you could answer it 2 or 3 different ways. I try to be very straightforward. So the practice test should give you a feel for that. Now, remember, there is no credit for the practice test, and you can take it as often as you want, and you can see what you're getting wrong and what you're getting right. So, hope I've answered that question. Any other questions? Okay, then I'm going to let us out of here a little early today for a change, and I'll see you. I guess I'll see some of you in lab today. So otherwise, have a good weekend, and I will see you next.